Hi everyone, and welcome to the Chemistry, Biology and Math Revision Hub. Today we'll be going through the Pearson Edexcel International A-Level Biology Scientific article focusing on question 8 for the June 2025 exam. Let's begin with Article 1. The title of the first article is Risk of Heart Failure May Increase with Age Due to Y Chromosome Loss. The Y chromosome involved in sex determination mysteriously disappears from some men's immune cells as they get older, and that could be fatal. The immune cells of many older men lose their Y chromosomes, and this may contribute to men having a higher rate of heart disease than women. Health risks from Y chromosome loss have long been suspected, but evidence from animals and people now adds more support to the idea and may suggest a treatment for the damage caused to the heart. DNA is packaged into chromosomes, with the cells of most men and transgender women carrying one X and one Y, and those of most women and transgender men carrying two X's. Although some people have other combinations, it was discovered several decades ago that in some people born with XY chromosomes, a proportion of the immune cells have no Y chromosome, a phenomenon that becomes more common with increasing age. For instance, 40% of 70-year-old men have no Y in at least some of the immune cells found in their blood. The reasons for this are unclear, but it could be because the Y chromosome is small and carries relatively few genes apart from those involved in sex determination and sperm production, so the stem cells that produce immune cells can survive if they happen to lose their Y when replicating their DNA. If they lose any other chromosomes, however, the cells would be more likely to die. Loss of this Y chromosome in immune cells correlates with higher rates of health problems, including heart disease, cancer, and Alzheimer's disease. But it was unclear whether Y loss causes these conditions or if faulty DNA replication is behind both Y loss and the health issues, says Lars Forsberg at Uppsala University in Sweden. To find out, Forsberg and his colleagues used the CRISPR gene editing techniques to remove the Y chromosome from about 2/3 of the immune cells of male mice to mimic the phenomenon. These mice developed heart problems once they got to about 1 year of age, precipitated by their heart muscle becoming scarred. We show causality, says Forsberg. We can see that losing the Y chromosome in the blood causes disease in the heart. The researchers also looked at how Y chromosome loss affected men in a large ongoing medical study called UK Biobank, which tracks participants' health over time. They found that the more immune cells without a Y that the men had at the time of their enrollment, the higher their risk of dying from any type of heart disease over the following 12 years. For instance, men who had lost the Y chromosome in more than 40% of their immune cells had a 31% higher risk of dying from any circulatory disease during the study period. Scarring of heart muscle can contribute to some common types of heart disease, such as heart failure, says Forsberg. Further work on the mice uncovered clues about how immune cells cause heart scarring. When the animals were dissected, immune cells lacking a Y chromosome were found to have infiltrated heart muscle, triggering the release of an inflammatory signaling molecule called transforming growth factor beta. Treating the mice with an antibody that blocks this growth factor reduced the harmful effects of the loss of the Y chromosome, although it is too soon to conclude the same approach would benefit men who are losing their Y chromosomes, says Forsberg. He is co-founder of a biotech firm called Cray Innovation that is developing a blood test to tell people if they have loss of the Y chromosome in their immune cells. This is the best evidence I have seen for a direct effect of Y chromosome loss on a physiological process, says John Perry at the University of Cambridge. In 2019, Perry and his colleagues published work suggesting that a propensity for Y chromosome loss is caused by many genetic variants that raise the likelihood of DNA errors during cell division, which could be why loss of the Y chromosome is linked with cancer. Forsberg's team next plans to investigate whether people born with XY chromosomes experience different effects if they lose one of their Y chromosomes. Moving on to the second article, the title is Essence of Man, Why Size Doesn't Matter. The Y chromosome, which makes men male, has been shrinking for 180 million years, but there's more to this rotting husk than anyone suspected. There's nothing very macho about the Y chromosome. Even though it's what makes men male, the human Y, like its counterparts in almost all mammals, is tiny compared with its partner, the X chromosome. It's lost hundreds of genes, and if the Y continues to lose them, it could someday wink out of existence entirely. Claims of its impending demise are starting to look premature, however. Far from being a rotting husk, the modern Y, tiny though it is turning out to be a highly evolved and surprisingly important part of man's wider genetic endowment, responsible for far more than just maleness. It is easy to see why some biologists thought the Y was destined for oblivion. It is all on its own. There are two copies of all other chromosomes, which are basically containers for holding genes. Each copy acts as a backup for the other. The pairs line up and swap bits when organisms reproduce. Some offspring get landed with chromosomes full of damaged genes and are eliminated by natural selection, whereas others inherit undamaged copies and survive to reproduce. Way back in the evolutionary past, there was no Y, just a regular pair of chromosomes. Sex was determined by environmental factors such as temperature, but then a gene on a single chromosome mutated in a way that made any individual that inherited it male. At first, this proto-Y could still swap genes with its partner, the proto-X chromosome. About 180 million years ago, in the line of mammals that branched away from the platypus and echidna, a section of it containing the gene variant for maleness got flipped back to front. This section no longer lined up properly with the corresponding part of the proto-X, so damaged genes in this section could no longer be swapped for good ones. Beyond repair, further inversions put more and more of the Y beyond repair. The X was fine because females inherit two copies that can swap parts. The Y, however, started to lose bits because men have just one copy. The human version now has just 78 genes, far less than its original 600 or so. At this rate of decay, the Y ought to disappear altogether within 5 million years, as famously predicted a few years ago by Jenny Graves at La Trobe University in Melbourne, Australia. But there is growing reason to believe that what's left of the Y is here to stay. For one thing, even though it has lost almost all of its original set of genes, it has gained others. We now know that 61 of the human Y's 78 genes were not present before the first inversion took place. Almost all the new genes play a role in sperm production, making the Y a perfect home for them. There are often several copies of these genes too, so there are backups. An even stronger reason to think the Y chromosome has a bright future comes from the discovery by Daniel Bellot at the Whitehead Institute in Boston that its decay seems to have ground to a halt. His team compared the Y chromosomes of 8 mammals, human, chimp, rhesus macaque, marmoset, mouse, rat, bull, and opossum, to trace its evolutionary history. They found bursts of gene loss directly after inversions happen, followed by long periods of stability. In fact, not a single gene has been lost from the oldest part of the human Y in the past 44 million years. The remaining genes may simply be too essential to lose. A team led by Henry Kessmann at the University of Lausanne, Switzerland, surveyed the Y chromosomes of 15 different mammal species and one bird. They found that a chromosome linked with maleness evolved 3 distinct times, once in birds, once in the ancestor of the platypus and echidna, and a third time in the ancestor of all other mammals. The ancestors of the 3 Ys each started with different kinds of genes, but to Kessmann's surprise, all ended up with a stable set of the same sorts of genes, which is what Bellot's team also found. You play this evolutionary game with different sets of genes, and you get the same kinds of genes retained in each case. He says it's always the regulatory genes that remain. Why? When a gene is lost from the Y, males are left with one copy of the gene on their single X chromosome. That means less of the protein the gene codes for gets made, roughly half the usual dosage. Evolution can fix this in males by ramping up production from the single X, but then their female descendants get a double dose from their 2 X's. To keep gene output the same in the 2 sexes, despite this difference, females have evolved to inactivate one of their 2 copies of most genes on the X. Perhaps the amount of protein produced by the regulatory genes retained on the Y had to be so precisely calibrated that organisms couldn't survive the awkward intermediate stage when this workaround did not yet function perfectly, suggests James Turner at the MRC National Institute for Medical Research in London. Regulatory genes are particularly vital because they control many other genes. So important are the Y genes, in fact, that even during a man's lifetime, losing the Y in some tissues takes a toll. Chromosomes can be lost when cells divide, and men who lose the Y chromosome in their bone marrow, which happens in about 8% of elderly men, have a higher risk of cancer and die an average of 5.5 years younger than other men. From the article, here are effects of Y chromosome loss. Loss of the Y chromosome in blood cells, common in aging men, impairs immune cell function. These Y-deficient cells contribute to chronic inflammation and infiltrate heart tissue, releasing molecules that cause fibrosis, scarring of the heart muscle. This stiffens the heart and reduces its ability to pump blood, increasing the risk of heart failure. At the same time, the weakened immune response reduces the body's ability to detect and eliminate cancer cells. Chronic inflammation caused by Y-deficient immune cells can also promote tumor growth. Importantly, recent studies have linked Y chromosome loss to neurodegenerative diseases. Men with this chromosomal loss show a significantly higher risk of developing Alzheimer's disease, likely due to impaired immune surveillance in the brain and increased neuroinflammation. Before we begin with the potential questions, I'd like to make a quick disclaimer. Please note that I have not seen your actual exam paper. The information presented here is based solely on the content provided in the scientific article. I am simply breaking it down and creating potential questions to help guide your revision and preparation. The questions in your exam may not be worded exactly as they are here. However, the content covered in these questions and answers has a strong likelihood of appearing in some form during your exam. Let's begin with the potential questions. These are broken down into sections. Section 1. Effect of cardiac inflammation on heart function. Question 1. How does histamine contribute to inflammation? Here you could include that histamine is released by mast cells and basophils. It causes vasodilation, increasing blood flow to the area. It also increases capillary permeability, allowing immune cells to enter tissues. Question 2. Explain why heart function may decline as a result of immune cell infiltration. Your answer could include immune cells may release cytokines and enzymes that damage tissue. This can lead to scarring and inflammation of cardiac muscle. Damaged tissue is less able to contract effectively, lowering output. Question 3. Describe the effects of fibrosis, scarring on heart muscle function. Fibrosis replaces muscle tissue with non-contractile scar tissue. This reduces the elasticity and efficiency of the heart's contraction. It may lead to reduced stroke volume and heart failure. Question 4. How can chronic inflammation in cardiac tissue affect heart rhythm? You can say, inflammation can damage the conduction system of the heart. This may disrupt electrical signaling from the SAN to the ventricles. It can result in arrhythmias or irregular heartbeats. Section 2. Antibodies blocking inflammatory factors. Question 5. Explain how antibodies can reduce the effects of harmful inflammatory factors. You could say, antibodies can be designed to bind specifically to inflammatory signaling molecules like cytokines or growth factors. This prevents the inflammatory factor from binding to its receptor on target cells. As a result, the downstream inflammatory response, like tissue damage, is reduced. Question 6. Describe how using antibodies to block inflammatory molecules could protect heart tissue. Inflammatory molecules can lead to tissue damage and scarring in the heart. Antibodies that bind and neutralize these molecules prevent their interaction with heart cells. This reduces inflammation, fibrosis, and helps maintain normal cardiac function. Section 3. Muscle contraction. Question 7. Explain the role of calcium ions in the contraction of skeletal muscle. You can include calcium ions are released from the sarcoplasmic reticulum in response to an action potential. They bind to troponin, causing tropomyosin to move and expose binding sites on actin, forming cross bridges and initiating contraction. Question 8. How is ATP involved in the sliding filament theory? A model answer could say, ATP is hydrolyzed to provide energy for the power stroke. It also allows the myosin head to detach from actin. New ATP resets the myosin head for another cycle. Question 9. Explain why muscle contraction stops in the absence of ATP. Myosin heads cannot detach from actin. Calcium cannot be pumped back into the sarcoplasmic reticulum. This leads to sustained contraction and eventual muscle rigidity. Question 10. Compare the structural features of fast-twitch and slow-twitch muscle fibers. Slow-twitch fibers contain more mitochondria, more myoglobin, and a richer blood supply. Fast-twitch fibers have fewer mitochondria and less myoglobin, but a greater store of glycogen and anaerobic enzymes. These differences reflect their specialization for aerobic in slow-twitch or anaerobic in fast-twitch respiration. Question 11. Explain how fast-twitch and slow-twitch fibers are adapted for different types of physical activity. You can say slow-twitch fibers contract slowly and are fatigue-resistant, making them ideal for endurance activities like long-distance running. Fast-twitch fibers contract rapidly and generate greater force, but fatigue quickly, suitable for short bursts of activity like sprinting. Their metabolic and structural adaptations match their function. Question 12. What is meant by the heart being myogenic? Myogenic means that the heart muscle generates its own electrical impulses without nervous input. The sinoatrial node, SAN, initiates depolarization, setting the pace of the heartbeat. This allows the heart to beat rhythmically and continuously on its own. Question 13. Describe how the myogenic nature of cardiac muscle coordinates the heartbeat. The SAN generates an electrical impulse, causing the atria to contract. The impulse is delayed at the AVN, allowing ventricles to fill. It then travels down the bundle of His and Purkin fibers, triggering ventricular contraction from the apex upward. Section 4. Nervous system and impulse transmission to muscles. Question 14. Describe how neurotransmitters transmit a signal across a synapse. An action potential arrives at the presynaptic knob, causing calcium channels to open. Calcium ions bind to vesicles, triggering them to release neurotransmitters into the synaptic cleft. The neurotransmitters bind to receptors on the postsynaptic membrane, triggering a new impulse. Question 15. Describe how a nerve impulse is transmitted along an axon. A stimulus causes depolarization as sodium ion channels open and sodium enters the axon. The inside becomes more positive, triggering adjacent sodium channels to open, a wave of depolarization. The impulse travels along the axon. Repolarization follows as K+ ions exit the cell. Question 16. Explain how myelination affects the speed of nerve impulse transmission. Myelin sheath insulates the axon, preventing ion movement except at nodes of Ranvier. The impulse jumps between nodes via saltatory conduction. This increases the speed of transmission compared to unmyelinated fibers. Question 17. Describe how an action potential leads to muscle contraction in skeletal muscle. The action potential travels down the motor neuron and causes release of acetylcholine at the neuromuscular junction. This triggers depolarization of the muscle fiber and release of calcium ions from the sarcoplasmic reticulum. Calcium ions bind to troponin, exposing actin binding sites and allowing myosin heads to form cross bridges and initiate contraction. Section 5. Regulatory genes on the Y chromosome. Explain the role of regulatory genes on the Y chromosome and why they are conserved across species. You could say, regulatory genes control the expression of other genes, often influencing essential cellular processes. The Y chromosome retains a small number of highly conserved regulatory genes important for male development and fertility. These genes are preserved by natural selection because mutations in them can be harmful or lethal, reducing reproductive success. Section 6. Loss of the Y chromosome with age. Question 19. Explain why some men lose the Y chromosome in their cells as they age. During repeated cell divisions, errors in chromosome replication can occur. The Y chromosome is small and gene poor, so cells that lose it can often still survive and function. As men age, these replication errors become more frequent, leading to loss of the Y chromosome in a proportion of their immune cells. Section 7. Mutation. Question 20. Explain how mutations in DNA can lead to the loss of the Y chromosome in some cells. Mutations affecting genes involved in DNA replication or repair can cause errors during cell division. If such mutations affect chromosomal segregation or spindle attachment, The Y chromosome may not be properly distributed. Cells that lose the Y chromosome but retain essential autosomal genes may survive, especially because the Y carries relatively few critical genes. Question 21. Explain how some mutations do not affect the survival of a cell. Some mutations are silent. They change a DNA base but not the amino acid due to the degenerate nature of the genetic code. Other mutations may occur in non-coding regions, e.g. introns or regulatory sequences, and have no effect on protein function. If the altered protein still functions sufficiently, the cell can survive and function normally. Question 22. Explain how genetic variants of the Y chromosome are formed. Genetic variants arise through mutations, such as base substitutions, insertions, or deletions during DNA replication. These changes can occur spontaneously or due to environmental factors like radiation. During meiosis, the Y chromosome doesn't undergo recombination over most of its length. These mutations accumulate and are passed down unchanged from father to son. Section 8. Survival of some immune cells with Y chromosomal loss. Question 23. Why might immune cells tolerate loss of the Y chromosome? The Y chromosome does not carry essential genes for immune function. Other chromosomes may compensate for lost functions. The cells still perform basic immune roles. Question 24. How can loss of the Y chromosome in immune cells cause heart damage? You can say it leads to abnormal immune cell signaling, promotes chronic inflammation and scarring of heart tissue, reduces the heart's ability to function properly. Question 25. How can Y chromosome loss contribute to disease? Affects gene expression in immune cells, can trigger inflammation and organ damage, e.g. in the heart, linked to increased risk of cancer and shorter lifespan. Question 26. What does the survival of cells with chromosomal loss suggest about genetic redundancy? Some genes have duplicates or backup pathways. Cells can adapt to partial genome losses. Essential functions are preserved by regulatory systems. Section 9. Gene loss and retention by natural selection. Question 27. Explain how natural selection leads to the loss or retention of genes in a population. Genes that code for beneficial traits increase an organism's survival or reproductive success, so they are passed on more often and retained. Harmful mutations in genes reduce fitness and are selected against, leading to the gene being lost over generations. Neutral genes may persist or be lost unless they become beneficial or harmful under new environmental conditions. Section 10. Apoptosis. Question 28. What triggers apoptosis in cells? DNA damage, infection, or signals from other cells. The cell is broken into apoptotic bodies for phagocytosis. Question 29. Why is apoptosis important in development? It removes unneeded cells, e.g. between fingers in embryos. It shapes organs and tissues, prevents overgrowth or malformations. Question 30. Explain how apoptosis can lead to neurodegeneration. Apoptosis is a controlled process that removes damaged or malfunctioning cells. In the brain, excessive activation of apoptosis can lead to the loss of large numbers of neurons. This cell loss reduces brain function over time, contributing to neurodegenerative diseases such as Alzheimer's. Section 11. Imaging the brain. Question 31. Compare the uses of CT and MRI scans in brain imaging. CT uses X-rays to create cross-sectional images of the brain and is useful for detecting bleeding, tumors, or bone injuries. MRI uses strong magnetic fields and radio waves to produce high-resolution images of soft tissues. MRI provides more detail for detecting brain abnormalities like tumors. Question 32. Explain how functional MRI, fMRI, shows brain activity. fMRI detects changes in blood oxygenation linked to neuroactivity. Active brain regions receive more oxygenated blood, which alters the magnetic properties detected by the scanner. These changes are converted into colored maps showing which areas are active during specific tasks. Question 33. Describe how a PET scan works and what it can detect in the brain. A radioactive tracer, often a glucose analog, is injected into the bloodstream. Active brain cells absorb more of the tracer, which emits positrons detected by the scanner. PET scans measure metabolic activity and are used to detect functional abnormalities, such as those seen in Alzheimer's disease or brain tumors. Section 12, Control of Gene Expression. Question 34, describe the role of transcription factors in regulating gene expression. Transcription factors are proteins that bind to specific DNA sequences, promoters or enhancers. They either promote or inhibit the binding of RNA polymerase to the gene. This regulates the initiation of transcription and controls gene expression. Question 35. Explain how DNA methylation can lead to gene silencing. Methyl groups are added to cytosine bases near promoter regions. This changes the structure of chromatin, making it more condensed. RNA polymerase cannot access the gene, so transcription is prevented. Question 36. Describe how histone modification can influence gene expression. Histones can be modified by acetylation or methylation. Acetylation reduces the positive charge on histones, loosening their interaction with DNA and increasing gene accessibility for transcription. Methylation can either activate or repress gene expression, depending on which amino acids on the histone are methylated and how many methyl groups are added. Question 37. Explain how non-coding RNA can regulate gene expression. Some non-coding RNAs bind to complementary mRNA. This can block translation or lead to degradation of the mRNA. As a result, the corresponding protein is not produced. Section 13, Microarray and Bioinformatics. Question 38. Describe how a DNA microarray is used to compare gene expression in two tissue samples. Messenger RNA is extracted from both tissues and converted to complementary DNA using reverse transcriptase. The cDNA samples are labeled with different fluorescent dyes and applied to a microarray chip. Each spot on the chip contains DNA probes for specific genes. The intensity and color of fluorescence at each spot indicate relative gene expression levels in the two tissues. Question 39. Give two advantages of using microarrays in biomedical research and one limitation. Advantage: allows simultaneous analysis of thousands of genes in a single experiment. Helps identify gene expression differences between healthy and diseased cells. Limitation: only detects genes already represented on the array. It cannot discover unknown genes. Section 14, Gene Editing, Knockout and Silencing. Question 40. Explain what is meant by a gene knockout. A gene knockout involves removing or disrupting a gene so it cannot be transcribed or translated. This prevents the production of the protein encoded by that gene. Scientists use knockout models to study the function of specific genes by observing what changes when they are missing. Question 41. Describe how gene silencing can prevent a gene from being expressed. Gene silencing involves blocking the transcription or translation of a gene without changing the DNA sequence. This can occur via mechanisms like DNA methylation, histone modification, or non-coding RNA. As a result, the protein product of the gene is not produced. Question 42. Give one difference and one similarity between gene knockout and gene silencing. Knockout removes or disrupts the gene itself. Silencing prevents its expression without altering the gene sequence. Similarity: both stop the production of the gene's protein product. Both are used to study gene function. Question 43. Define CRISPR. This is a gene editing tool that uses a guide RNA and the Cas9 enzyme to make precise cuts in DNA. It allows scientists to add, remove, or modify specific genes in an organism's genome. Section 15. Genetic modification. Question 44. Define genetic modification and give one example. Genetic modification involves altering the genetic material of an organism by inserting, deleting, or changing specific genes. This is often done using recombinant DNA technology, for example, inserting the human insulin gene into bacteria to produce insulin for medical use. Question 45. Describe how a gene can be inserted into a bacterial plasmid. A restriction enzyme cuts the plasmid and the target gene at specific sequences, producing complementary sticky ends. The target gene is inserted into the plasmid using DNA ligase. The recombinant plasmid is introduced into bacteria via transformation, e.g. heat shock or electroporation. Question 46. Explain one advantage and one ethical concern of genetic modification in agriculture. Advantage: GM crops can be made resistant to pests, increasing yield and reducing the need for pesticides. Ethical concern. Some people are concerned about the long-term ecological effects or health impacts of GM foods. There are also concerns about the control of food supply by biotech companies. Section 16, Parkinson's disease. Question 47, what causes the symptoms of Parkinson's disease? Parkinson's disease is caused by the death of dopamine-producing neurons in the substantia nigra of the brain. This leads to reduced dopamine levels in motor pathways. As a result, muscle control and coordination are affected. Question 48. Explain how L-DOPA can be used to treat Parkinson's disease. L-DOPA is a precursor to dopamine that can cross the blood-brain barrier. Once inside the brain, it is converted into dopamine. This increases dopamine levels, improving motor control and reducing symptoms. Question 49. Describe two motor symptoms of Parkinson's disease and their biological cause. Symptoms include muscle rigidity and tremors. These result from decreased dopamine levels. Dopamine is needed for smooth and coordinated muscle movement. Section 17, depression. Question 50, describe the biological explanation for depression. Depression is linked to low levels of serotonin in the brain. This can be due to reduced production, release, or receptor sensitivity. Low serotonin affects mood regulation and emotional balance. Question 51, how do SSRIs work to treat depression? Selective serotonin reuptake inhibitors block the reuptake of serotonin in the synaptic cleft. This increases the concentration of serotonin at synapses. Higher serotonin levels improve communication between neurons and help stabilize mood. Question 52. Give one reason why not all patients respond to SSRI treatment. The cause of depression may involve other neurotransmitters, e.g. dopamine or noradrenaline. Some individuals may have receptor level issues that SSRIs cannot correct. Genetic variation can affect drug metabolism or response. Section 18, nervous control of the heart. Question 53, describe how the medulla oblongata controls heart rate. It receives input from baroreceptors and chemoreceptors. It sends impulses via the sympathetic or parasympathetic nervous system. This alters the frequency of impulses from the SAN. Question 54. How do the sympathetic and parasympathetic systems differ in their effects on the heart? The sympathetic nervous system increases heart rate and force of contraction. It releases noradrenaline onto the SAN. The parasympathetic system reduces heart rate by releasing acetylcholine. Question 55. What is the role of the sinoatrial node, SAN, in heart control? It generates electrical impulses that initiate heartbeats. It sets the pace for the heart, known as the pacemaker. These impulses spread across the atria, causing them to contract. Section 19, hormonal control of the heart. Question 56, explain the effect of adrenaline on the heart during stress. Adrenaline binds to receptors on cardiac muscle. This increases the frequency and force of contraction. Heart rate and cardiac output increase to supply more oxygen to tissues. Question 57. Describe the hormonal changes that occur in response to low blood pressure. Adrenal glands release adrenaline and noradrenaline. These hormones cause vasoconstriction and increase heart rate. This raises blood pressure back to normal. Question 58. Describe the process of transcription in protein synthesis. RNA polymerase binds to the promoter region of a gene and separates the DNA strands. It uses one strand as a template to assemble a complementary strand of pre-mRNA using free RNA nucleotides. Transcription continues until a stop signal is reached. The pre-mRNA is then processed, like intron removal, to form mature mRNA. Question 59. Explain how the sequence of bases in mRNA determines the structure of a protein. The base sequence in mRNA is read in triplets called codons. Each codon codes for a specific amino acid. The sequence of codons determines the order of amino acids in the polypeptide, which then folds into a specific three-dimensional protein structure. Question 60. Describe the role of tRNA in the process of translation. Each tRNA molecule has an anticodon that is complementary to an mRNA codon. It carries a specific amino acid to the ribosome. The amino acids are joined by peptide bonds to form a polypeptide chain. Question 61. Explain the process of RNA splicing and its role in gene expression. Introns, non-coding regions, are removed from pre-mRNA. Exons, coding regions, are joined together to form mature mRNA. This ensures that only coding information is translated into a protein. Question 62. How can alternative splicing lead to different proteins being produced from the same gene? Different combinations of exons can be joined during RNA splicing. This results in mRNA molecules with different coding sequences. Each variant is translated into a different protein. Section 21. Hormones and cell function. Question 63. Explain how chemicals such as hormones or drugs can affect heart function. Chemicals like adrenaline bind to receptors on cardiac muscle cells. This increases calcium-ion permeability, which enhances the strength and rate of contraction. Other chemicals, e.g. acetylcholine, can slow heart rate by increasing potassium-ion efflux, reducing excitability. Question 64. Explain how hormones can affect the function of cells that carry mutations. Hormones bind to specific receptors on or in target cells, triggering intracellular signaling pathways. In mutated cells, the response to hormones may be altered if the mutation affects hormone receptors or signaling proteins. This can lead to abnormal gene expression or uncontrolled growth, such as in hormone-sensitive cancers. Question 65. Explain how the loss of the Y chromosome in some cells may contribute to the development of cancer. The Y chromosome loss may result in disrupted gene regulation, including genes involved in cell cycle control. This can impair immune surveillance, allowing abnormal cells to survive and divide. Accumulation of further mutations in such cells may lead to uncontrolled proliferation, a key feature of cancer. Section 22. Clinical trials. Question 66. Describe the purpose of Phase 1, 2, and 3 clinical trials. Phase 1. Test the drug on a small group of healthy volunteers to assess safety, side effects. Phase 2. Test the drug on a small group of patients to evaluate effectiveness and side effects. Phase 3. Compares the drug to existing treatments in a large group of patients to confirm effectiveness and monitor long-term effects. Question 67. Explain why double-blind and placebo-controlled methods are used in clinical trials. Double-blind means neither the participants nor researchers know who receives the treatment, or placebo, reducing bias. A placebo allows comparison with a group not receiving the active drug. This ensures that observed effects are due to the drug and not psychological or environmental factors. Question 68. Why is randomization important in clinical trials? Randomly assigning participants to treatment or control groups reduces selection bias. So this brings us to the end of the article. Thank you for being with us. See you in the next video. Bye-bye.